students, I hope all of you are doing very well. So, we are continuing our understanding in immunity, immunology, how you can produce various types of immunological products like vaccine, anti-serum, monoclonal antibodies and other things. So, let me continue same understanding in our unit 3 of pharmaceutical biotechnology. So, today I am going to tell you something very interesting technology called Hybridoma technology. This is very important in the production of the monoclonal antibodies. So, today I will tell you, let me take the ink, yeah. So, today I will tell you, at first I will just recall our previous understanding of antibodies. I will tell you how antibodies different with vaccine, monoclonal antibodies, hybridoma technology, history of hybridoma technology, background, how it is actually working, steps what are involved in hybridoma technology, and then I will give you a summary. So, let me continue. First, let me tell you about the antibodies. So, you are aware that antibodies are playing a big role in case of our to boosting our immunity. If a foreign substance may enter in our body, then antibodies are actually come to the picture. SARS-CoV-2 coronavirus, I have tried to bring you the most common example, because COVID-19 right now already created a huge problem in all over the world. So, SARS-CoV-2 is responsible for COVID-19. So, if a SARS-CoV-2 actually enter in your body, and in your body if you have already some that is if you already taken some vaccine, so in your body there will be some antibodies. Now, how antibodies will stop the spreading of SARS-CoV-2? Very interesting. You can see these are the various spike proteins. So, your antibodies will bind with those various proteins, and it will cover, antibodies will cover and tear all those various that is a surface of this virus. The best part of our antibodies is that this spike protein, and in our human cell, that is in our, especially in our lung, we have ACE2 receptor, ACE2 receptor. Okay. So, the spike protein usually actually bind here. Due to effectivity of antibodies, antibodies will be go there and it will bind with all these that is a outer surface of the spike protein. Due to antibodies, this SARS-CoV-2, it will be not able to make a complex or make a bind with your receptor like ACE2, and through that antibodies are actually saving your yourself from COVID-19. Okay. So, this is a typical example how that is antibodies are actually saving us from SARS-CoV-2. This concept is applicable in almost all kinds of your these things, vaccines. Okay. Now, let me tell you one by one. So, one is antibody therapy and another is vaccine. So, I am very much interested to tell you the differences between these things, because a lots of time I have seen the students are actually get confused. Okay. They think the antibody therapy and vaccine both are same. No, both are not same. When you will give vaccine in a person's body, his immune system will be trained against that vaccine. Vaccine is containing antigen. His immune system will be get trained against that antigen, and it will produce antibody. Okay. In future, the virus will enter in the body, his antibody will come to the picture, it will take the action. So, at first you will be give the vaccines, and then the antibodies will be developed. So, that is why in antibody therapy what we are giving, in antibody therapy a protein that binds to the virus and stop it infecting you. So, means somebody already got vaccinated with COVID-19 or any of the diseases, already he will have some antibody. If you take out some antibody, then if you somebody suffering from COVID-19, if you give that antibody to that patients, then the patient will be able to survive because of the antibody therapy. So, what is it? A protein that binds to the virus and stop it from infecting you. Vaccine, a piece of virus your immune system can recognize. First we give this, then antibodies are produced, then we may take the antibody from the person's body, then we may give that antibody to the other person who is actually suffering from that virus. What is it for? Treat those are already infected with COVID-19 and train the immune system, whatever I was mentioning you, to fight against COVID-19 virus. How fast does it work? As soon as it is given, and here it will take some time for the development of the antibody, like 1 to 2 weeks. How long does it production is last? 6 to months, and in this case year to lifetime. Okay, year to lifetime, but you need some booster dose. For example, SARS-CoV-2 COVID-19, either Covaxin or Covishield, you have to take two different doses. Okay. So, in recent times we have seen after the advancement of Delta variant, lots of scientists are saying that 2 doses will be not sufficient, we have to take another dose as a booster dose. Okay. So, that you can get the lifetime that is a vaccination. Now, come to another very interesting concept, monoclonal antibodies. So, you have seen in my previous slide that how the antibodies are actually that is going and bind with the virus surface, and it is actually creating a big complex, we call it as opsonization. So, monoclonal antibodies are also working in the same concept. So, for example, this you can see this is a cancer cell. Cancer cells are supposed to be die. So, monoclonal antibodies are actually going and binding with its various receptor. So, this Y shaped protein will be go and it will bind like this, you can see it is binding here. Okay. So, antigen protein on the cell that cause the immune cell to respond. Monoclonal antibody lock onto the antigen, this cause the immune system to attack on cancer cells. Yeah, another thing I will to tell you, when your all the antibodies are actually that is a covered now, then when it will covered by your various antibodies, then your immune system cells like cytotoxic T cell, killer T cell, they will come to the picture, they will engulf and other dendritic cell, neutrophil and other that is cells, macrophage etcetera, they will engulf it and remove this from the body. So, monoclonal antibodies developed in laboratory to help our body to fight infection. Nearly 100 of the monoclonal antibodies are actually approved for the various diseases like cancers and also autoimmune diseases. Monoclonal antibodies are also being studied for the treatment of the prevention of COVID-19. Okay. Now, let me tell you how they are works, similar process whatever I was tried to tell you at the beginning of my lecture with SARS-CoV-2 example. So, monoclonal antibodies will bind their target, you can see these are actually covered. Okay. These are all the monoclonal antibodies, this Y shape, always remember that antibody will be in Y shape. So, you can see these are Y shape, it is actually covered, and after that once attached, when monoclonal antibodies will attach now, then this virus will become harmless, because it is already covered, it cannot react with anything, it cannot react or it cannot bind with any of the other that is cells. You can simply that is a call that monoclonal antibodies are like police who are arresting. When a criminal is actually got arrested by the police, then the criminal cannot do any kind of crime in public. So, this virus or this cancer cell, it is actually arrested, you can see it was actually completely that is covered by our that is monoclonal antibody. So, it is like arrested. So, it will not able to harm. These are few example of various kinds of monoclonal antibodies. You can see in every name at the end MAB is actually written. You should always remember as a student of pharmacy, when there will be at the end there is a written MAB, it will be a monoclonal antibody. I have written in detail, you may read in your free time. I am not going to discuss in individual name. So, you can see maximum of the cases maximum of these cases, these are actually cancerous that is for cancer this monoclonal antibody is actually developed, and if you see almost every 2 or 3 years new monoclonal antibodies actually being approved. Here are some other examples, again you can see for cancer. So, that is why you have to understand, if you want to shine your career in cancer therapy, you have to focus on monoclonal antibody, okay, because this is the trending, and in future you will be observing that we will be able to eradicate cancer from the world. Yes, you are listening correctly, we will be able to eradicate cancer from this world completely. Why? Because of the great technology or great advancement of hybridoma technology. By using hybridoma technology, we can make monoclonal antibodies, and monoclonal antibodies are very effective on various cancer diseases. That is why every year you can see every 2-3 year lots of that is monoclonal antibodies actually approved for cancers. You can see lots of cancers, these are all cancers, okay. You can see cancers. So, that is why it is very much important. Another thing I like to mention you, because sometimes you oftenly get that is confused between the polyclonal antibody, monoclonal antibody, and another thing called monoclonal antibody cocktail. Last year, if I am not wrong, recent times when US election was going on, US presidential election, at that time Donald Trump was infected with COVID-19. The doctors were given him a very strong dose to him. It was not public at that time that what was actually given. He was actually given a monoclonal antibody cocktail. Due to that, he recovered very fast. I think probably 3 to 4 days, if I am not wrong. So, polyclonal antibody, monoclonal antibody, and monoclonal antibody cocktail. In polyclonal antibody, there will be poly means multiple. You can see there are multiple various types of your these things antibodies are present. In monoclonal antibody, only one type of antibodies will be given. In cocktail, you will be observing that yes, there are multiple various types of antibodies actually given. This is how monoclonal, polyclonal, and cocktails are actually working. Now, I have told you lots of good things about this monoclonal antibody. Now, how you can develop or how you can make? You see, the most easiest way of getting this monoclonal antibody is taking the antibodies from the patients who already survived. Somebody was infected with COVID-19, he is now very well in his body, antibodies are present. If you may take those antibodies or from his body, from his that is blood. So, this will work very nicely in COVID-19 patients. But this is not a very simplest process, it is a very cruel or hard process, difficult process. That is why we need another technique or new technique by which we can make monoclonal antibody in our lab or in our industry scale production. That is why hybridoma technology has come to the picture. So, it is an innovative technology in biotechnology. It is a hybridization technique which is used to produce antibody producing hybrid cell. And these hybrid cells are produced by fusing genetic material of B lymphocytes with tumor cell. Okay, you may see the word fusing genetic material of B lymphocytes with the tumor cell. I will tell you more how it is actually working. The antibodies produced by monoclonal antibodies are as they are all a single specificity. For a certain, it is only single specificity to work. Presence of B lymphocyte genetic material helps in antibody production, whereas capacity to divide indefinitely in the cell culture due to the presence of the cancer tumor cell. Now, you might be observing that sir, in this slide you have mentioned tumor cell, in previous slide also you have mentioned tumor cell, but sir, as far as that is we know, cancer cells or tumor cells, these are not good, but you are using tumor cell here. Yes, you are listening correctly. This is, I can say, only one area where we can use tumor cells for our that is good purposes, for the beneficial of our entire humanity. You see what we are doing here. Let me again go to the previous slide. We are actually taking the genetic material of B lymphocytes. B lymphocytes are doing what? They are actually forming antibody. Cancerous cell or tumor cell, what is their activity or how they will work? Their main activity is they will rapidly grow, fast grow. You cannot stop or these cancerous cell or tumor cells, their growth is actually so much rapid. Okay. That is why we are actually fusing with those this both the that is B lymphocytes genetic material and tumor cells, so that when B lymphocyte cells are actually present in tumor cell will grow, the production will be very fast. I will tell you more, do not worry, I will tell you more with detail that how you can that is do this kinds of task. Let me tell you some basic history. So, the production of monoclonal antibody was invented by Caesar Milston and George JF called her in 1975. Okay. Then it was they pre-programmed B lymphocytes to respond to a single type of antigen or antigenic detriment. Therefore, they produce single type of antibody specific to the specific antigen. Okay. They have just done some experiment and they are successful. They tried to induce with some that is a antigen in that person's body, and then they have found that yes, the antibodies are being produced. The term hybridoma technology actually coined by Leonard Herzenberg during the subvertical Caesar's Milstein's laboratory in the same time, almost 1976 and 75. You can see 75 they have first done the experiment, and in the next 2 years they have coined this name that is a hybridoma. And let me tell you some more background that how it is actually working, because when an antigen actually react with B lymphocyte receptor, the lymphocytes divide very rapidly and produce a clone of B cells. All these B cells produce antibodies against that specific antigen, or we call it as a clonal section. Okay. So, you have some B cells or B lymphocytes. So, when some antigen you have given, for example, I am writing as an X, this is antigen. When your this X antigen it actually reacting with your B lymphocyte receptor, this B lymphocyte they will divide. They will divide and they produce clone of B cells, and these B cells will produce antibodies like this Y. Okay. They will produce a lots of antibodies. Those antibodies will be go here and it will surround this antigen or virus. Okay. So, that is why that is these things, these antibodies are actually important. Our main target is to identify this antibody. If I tell you more, the B lymphocyte cell produce one type of antibodies which are specific to only one type of antigen or antigenic detriment, but fully different antibody producing B lymphocytes known as plasma cell, and they do not actually divide, and when cultured. You see, you cannot culture the plasma cells, okay, or you cannot that is develop these things in your lab. That is a problem in terms of antibody. That is why you are actually focusing on hybridoma technology. Now, what is this and how we can use this technology? So, for example, you have one rat, okay. You have to bring that antigen to that rat, we call it as immunization. So, what will happen? We have seen in our this slide, when you are actually exposing some antigen with your B lymphocytes. So, they will divide themselves one by one, and then what they will do? They will develop some antibodies. So, obviously, this is very common in that rat's body. B lymphocytes and other things will be developed, antibodies will develop. So, you have to isolate the B cells from that rat. Okay. Then you have to take some myeloma cells, fusion partner. Okay. You have to take some myeloma cells, those are actually cancerous cells, simply you can say, and then you have to fuse those things. It will become like this. This complex we call it as a hybridoma. Clear? This complex we call it as a hybridoma. Then we have to select some het medium, and in that het medium we have to do some screening, and we have to expand that activity, and then we will be getting some monoclonal antibody. This monoclonal antibody you can give to the patient when he is this patient actually gets suffered from the similar kinds of antigen like viruses, etc. Let me tell you step by step how it is actually happening. So, what I have mentioned here in this slide, same thing I have tried to mention here, and step by step I want to tell you, because if you do the experiment in your lab, it will take some time, because some lengthy process is actually associated here. So, you have to take your rat, you need some myeloma cancerous cell. Okay. When you will give some antigen, some B lymphocytes will produce, you have to take out these B lymphocytes, and then you have to fuse with your this myeloma cell. Then you have to do for the head selection, then the hybridoma will form, then you have to do some assay, and then your cloned hybridoma will be produced. You have to culture, and then when it will be given to the rat or other kinds of animal, it will having it will work as an antibody. So, step by step if I tell you, the first step, mouse will be actually immunized, what I was telling you, and giving the antigen infection injection against the monoclonal antibody have to produce with an adjuvant, and this will be followed by the booster dose of antigen. After 72 hour, spleen will be collected in the mouse, because highest number of the your B cells will be produced in the spleen. This is the if I tell you more, and then a free cell suspension medium of the spleen actually prepared using sterile serum free medium followed by the centrifugation, because we have to separate those things, and all these processes are applied for the extraction of the plasma cell from the spleen. So, this is the step 1, this is the first step where you have done these kinds of activity. Step number 2, next step you have to maintain the plasma cell in the cell culture for at least 1 day, okay, before fusion. Before complete fusion you have to keep for some time. It should be ensured that the cells are in the early phase of growth at the time of the fusion. Then the myeloma cells are selected based on the criteria like these cells themselves should not produce antibodies, and also they should contain generic markers like HGPRT, and after that the genetic marker helps in the selection of the resulting of hybridoma cells. Then in step 3 you have to do the fusion. At the end of the fusion, both myeloma and spleen cells should be collected, then mix in an appropriate ratio. Okay. This myeloma cells and your this you have to take different ratio, like for example, that is a 5:1 or 2:2. You have to select based on your parameter. Following the mixing, the cells are centrifuged into a loose plate, and then supernatant will be removed, and the plates actually mixed in the polyethylene glycol for 3 minutes. In doing so, plates will be broken up into uniform small clumps. Then in step 4, following the infusion, dilute the cells in a serum free medium slowly to reduce the osmotic disruption of the fused cell. Then centrifuge the cells in response to the het medium, whatever I was actually trying to tell you here, and after that het medium containing the fused cell allowed to incubate in carbon dioxide incubator for 3 to 4 days. Okay. And then in step 5, the mixture of the cell population is then cultured in a selective media known as het medium along with the drug amino peptide. Then AGPRT myeloma cell cannot divide in the het medium due to the presence of this drug, and the specific antibody producing B lymphocytes are then able to divide continuously in the medium, and therefore, eventually they die. In step 5, only hybridoma cells have got the ability to divide and proliferate into the het medium, because genome of B lymphocytes makes them AGPRT positive, and genome from the myeloma cells they can divide indefinitely. That is the main thing whatever I was trying to tell you in earlier, why we are using the tumor cells, because we have taken the advantage of both the that is a tumor cells in our and our that is a B lymphocytes. Okay. Now, these things will be working very properly. Thus, only hybridoma cells or your fused cells are selected using the het medium. That is the main reason why we have actually used. Here is the summary. Again, I am tried to tell you in a most simplest way that how our this hybridoma technology is actually working. I hope after discussing a long part, now this picture will be very much clear to you. Let me again tell you that you have to take one rat, and for that disease for which you want to make monoclonal antibody, you have to, for that disease you are going to identify some monoclonal antibody, some part of if it is a virus related disease, some part of that virus you have to inject. Simply you have to inject some antigen associated with that particular disease. If it is a cancer or some other disease or such kind of disease, which disease can be treated with monoclonal antibody, you have to take the antigen. Then when we will give the antigen, then B lymphocytes will produce, then B cells, and you have to take some myeloma cells, you have to make some hybridoma, then you have to do for the het selection, you have to do some ELISA test if it is required, and then you have to do screening, and antibody producing hybridoma clones will be ready, and then at the end you will be getting monoclonal antibodies, and you can take those things for your work. So, that is it from my side. I hope you got a lots of information about how hybridoma technology is nowadays revolutionizing our this field called monoclonal antibody development, and lots of diseases right now we can solve through monoclonal antibody. So, that thank you all of you for joining my today's lecture. We shall meet in our next lecture. I will tell you more in particular that our understanding of immunity, and in future you should try to do something very good in this field, that is hybridoma technology. So, thank you all of you, thank you, God bless you all.